welcome to this class on neuroscience of human movement. In today's class, we will be discussing about disorders of the neuromuscular junction. So, in the previous class, we discussed neuromuscular junction. Let us briefly review that concept and then discuss the various disorders. So, in today's class, we will be talking about disorders of the neuromuscular junction. Specifically, we will discuss two autoimmune disorders, Lambert-Eaton syndrome, myasthenia gravis, and then we will be discussing the role of toxins in affecting the function of neuromuscular junction. There are several such toxins. There is botulinum toxin, there is hemicolinium, vesamical, tetanus, tuberculin, and other toxins whose mechanisms and actions we do not discuss in this class, right. So, let us briefly review what happens with the neuromuscular junction. Before we discuss neuromuscular junction, we will have to remind ourselves of how acetylcholine is synthesized or the life cycle of acetylcholine. Let us remember that acetylcholine is synthesized by choline acetyl transferase by the reaction of acetyl coenzyme A and choline in the presence of choline acetyl transferase. It is sometimes called CAT or sometimes called CHAT. Choline acetyl transferase is the protein that takes acetyl coenzyme A and choline and makes acetylcholine and packs them into bags called vesicles. We said that yesterday. And now acetylcholine itself is broken down into acetate plus choline by what are called as esterases. In this particular case, that is acetylcholine esterase. Okay. So, acetylcholine esterase breaks acetylcholine into acetate and choline, and choline acetyl transferase brings acetyl coenzyme A and choline together to make acetylcholine. So, this is the life cycle of acetylcholine. So, also remember acetylcholine esterase, when it breaks acetylcholine into two parts, basically into acetate and choline, part of this choline is reuptaken back into the presynaptic terminal. So, where is this acetylcholine esterase acting? At the synaptic cleft, ACH esterase. Acetylcholine esterase is acting at the synaptic cleft where it breaks acetylcholine into choline plus acetate. The acetate group escapes the system, whereas choline is reuptaken back to the presynaptic terminal by choline by what are called as, so choline is reuptaken to the presynaptic terminal by what are called as choline reuptake mechanisms. Okay. So, there are mechanisms to bring choline back into the presynaptic terminal, and this choline will be recycled by choline acetyl transferase to make more acetylcholine. So, this is the life cycle of acetylcholine, and so this acetylcholine is packed into bags called vesicles, such that whenever one vesicle is opened, all the molecules of acetylcholine in that vesicle are released. So, the idea that acetylcholine release happens in discrete quanta, where each quanta describes one vesicle, was first described by Bernard Katz, and for that he received a Nobel Prize. We saw that in previous class. ACH is released in discrete quanta, not continuously, and the amount of this release is related to the depolarization or the amount of calcium that is entering the cell due to depolarization or due to action potential. So, when an action potential arrives here, the calcium channels open, letting in a lot of calcium, and synaptotagmin senses the presence of this calcium. Synaptotagmin is the calcium sensor on the vesicle. So, synaptotagmin senses the presence of this calcium and moves the vesicle closer to the cell body by the action of synaptobrevin and syntaxin, as shown in the next this. So, the action potential arrives here and calcium is detected by synaptotagmin, and it moves the vesicles closer to the cell membrane. By the action of docking proteins syntaxin and synaptobrevin, the vesicle fuses to the membrane and then opens up on the outside, releasing the neurotransmitter by a process called as exocytosis. We saw this in the previous class. Now the question is, what are the various things that could go wrong and what would be the effect of these problems? That is the question. First, we start out with autoimmune disorders. There are two specific cases that we will discuss in the context of neuromuscular junction. One is, for whatever reason, the immune system wrongly believes or wrongly labels the voltage gated calcium channels at the presynaptic terminal as an invader, as an outsider. Then what happens, the immune system attacks the voltage gated calcium channel, destroying them, preventing their function, right. So, what happens, so when this calcium channel is blocked, say for example, when this calcium channel is blocked or its function is reduced, action potential will happen in the presynaptic neuron and it will reach the voltage gated calcium channel, but since the voltage gated calcium channel is not functioning, it will not open. Since calcium is not entering the cell, synaptotagmin will not sense the presence of calcium. So, there will be no exocytosis. So, essentially acetylcholine will not be released. What is the effect? The effect is basically weakness and inability to perform voluntary movements, etc. So, this situation is called as Lambert-Eaton myasthenic syndrome. Lambert-Eaton myasthenic syndrome is due to blockage of calcium channels by the immune system in an autoimmune disorder. Usually this happens as a side effect, as one of the fallouts, as one of the problems associated with cancers, or in many cases, in about greater than 50% of Lambert-Eaton myasthenic syndrome cases are due to cancers. Okay. But there are other variants of this disease that also happens without a cancer. Okay. In many cases, this is due to side effect or one of the other effects of cancer. What else could go wrong? The other thing is when the nicotinic cholinergic receptors on the postsynaptic muscle side. So, this is the postsynaptic muscle membrane. Here there is the nicotinic cholinergic receptors, right, that open whenever acetylcholine comes and binds to these receptors. These channels open, causing an influx of sodium in the muscle membrane. It turns out that for whatever reason, if the immune system wrongly labels or wrongly believes that the nicotinic cholinergic receptors are if for whatever reason the immune system wrongly believes that the nicotinic cholinergic receptors are the outside agent, then it attacks the nicotinic cholinergic receptors, causing basically no more function. So, if the nicotinic cholinergic receptors are attacked by the immune system, then when they are destroyed. So, if the nicotinic cholinergic receptors are labeled as outside agents by the immune system for whatever reason, then what happens is that the nicotinic cholinergic receptors and their functions are blocked. So, acetylcholine that is released from the presynaptic terminal cannot attach to the receptor. So, when it is not able to attach to the receptor, what happens? The sodium will not enter inside the postsynaptic muscle cell, thus causing weakness and paralysis, etc. So, in the case of myasthenia gravis, what happens is action potential enters. In the case of myasthenia gravis, action potential reaches the synaptic end bulb and calcium is released into the presynaptic terminal, and this calcium is detected and the vesicles start moving and vesicles actually fuse and exocytosis of acetylcholine actually happens, but this acetylcholine cannot attach to any point on the postsynaptic terminal. So, because of that reason acetylcholine remains here, but it cannot attach to the receptors. So, sodium cannot enter inside, causing weakness and other problems that come with this disease, myasthenia gravis. Okay. Now, let us discuss the role of neurotoxins and how they affect the function at the neuromuscular junction, right. So, there are several things, several places. Let us remember choline is entering inside the system from the choline reuptake system, right, entering from the extracellular fluid to the presynaptic terminal via the choline reuptake system, right. If the choline reuptake system is somehow compromised, then you will have no more choline entering the system. Let us remember only about 50% of the choline is reuptaken and the other 50% is not reuptaken. For choline acetyl transferase to produce, so choline acetyl transferase basically combines acetyl coenzyme A and choline to produce acetylcholine. This is what we have seen. If this ingredient in acetylcholine is lesser, or if choline is reduced by 50%, essentially what will happen is acetylcholine cannot be produced, right. After some time acetylcholine cannot be produced. So, that action is caused by the toxin hemicholine. Hemicholine basically blocks the choline reuptake system from reuptaking choline. This will lead to a situation where choline cannot enter the system and whatever choline is already present in the presynaptic terminal only can be used. But essentially what will happen is after some time all the choline will be taken by choline acetyl transferase to make acetylcholine, and all the acetylcholine would be released into the synaptic cleft, right. After some time there will be no more choline, or there will be a very small amount of choline that is remaining that can be used by choline acetyl transferase to make acetylcholine. Essentially this will reduce the amount of acetylcholine in the presynaptic terminal, right. Here no more acetylcholine can be manufactured by choline acetyl transferase there, because there is no more raw material which is choline. Now, another thing that could go wrong is the case of what happens with the toxin vesomicol. Now, vesomicol, what it does is it causes vesicles to go empty by the following process. Essentially it does not allow the choline acetyl transferase to pack acetylcholine into bags. Now, that is essential, right. Acetylcholine will be remaining distributed, but they cannot go outside by themselves because there is no acetylcholine channel, right. They cannot go out like that. They have to be packed in bags, or they have to be packed in vesicles by choline acetyl transferase, and these vesicles must be in a position to detect the presence of calcium and then move and fuse with the membrane so as to exocytose acetylcholine. This is the mechanism through which acetylcholine can actually leave the presynaptic membrane, is it not? When acetylcholine is not packed into the synaptic vesicles, then it is not possible for acetylcholine to be released from the presynaptic terminal to the postsynaptic terminal. So, this chemical vesomicol actually prevents packing of acetylcholine into the vesicles, right. So, this is the other situation. Botulinum toxin is one of the most potent neurotoxins found, if not the most potent neurotoxin. So, how is it produced? Botulinum toxin is actually produced by the action of Clostridium botulinum, which is a bacteria, an aerobic bacteria that produces this very powerful toxin. There are several variations of this toxin, there are several forms of this toxin, but all of them have almost similar actions on the neuromuscular junction. What it does, how is it synthesized, how is it produced, right. Clostridium botulinum is an anaerobic bacteria and it produces botulinum toxin. Turns out that this is found in large quantities in honey, for example, which is why it is not a good idea to give honey to infants, right. Infants do not have the good bacteria or the healthy bacteria in their digestive tract that can fight the invaders like botulinum or Clostridium botulinum and other such dangerous bacteria, right. So, infants when they get honey, right, it is possible for Clostridium botulinum to produce botulinum toxin in their system, and since infants do not have the healthy bacteria to fight Clostridium botulinum will colonize the digestive tract of these infants and basically start producing botulinum toxin in relatively large quantities, and actually the tolerable limits is slightly above 1 nanogram per kilogram of body mass. So, we are talking about relatively small quantities, which is why I said this is one of the most potent neurotoxins, right. So, since infants are about a few kilograms, like 3-4 kilograms we are talking about, if they are given honey, they can only tolerate up to 3 nanograms, which can be easily produced by this bacteria, right. So, what this causes in infants is a condition called botulism. So, the infants eyelids will droop and the face will drop and the body will become limp, the eye and hand and leg movements will become very minimized, right. So, that is the symptom. Fortunately, it is possible to save these children from botulism if immediate action is taken. Okay. How does botulinum toxin itself work? That is the question. So, the release of acetylcholine is caused by the action of synaptobrevin and syntaxin, which basically form a part of a group of proteins called as snare proteins. Okay. Botulinum toxin, what it does is it prevents the ability of these snare proteins to interact. Basically, it cleaves the snare proteins. Then what will happen? Then the vesicles can no longer fuse with the membrane and exocytose the acetylcholine. So, vesicles will be present, they will have acetylcholine, but they will not be able to fuse and release acetylcholine into the synaptic cleft, essentially leading to a situation where all the acetylcholine is present in the presynaptic terminal, no acetylcholine is released in the synaptic cleft. So, the receptor is not able to sense the presence of acetylcholine and send in sodium, so that force can be produced. Essentially, this leads to a situation where there is weakness or paralysis. In many cases, this leads to total blockade, and in some cases it also leads to death, right. It turns out that there is a very crucial, very critical clinical use for botulinum toxin. If used in relatively controlled and small quantities by surgeons, it is possible to release tight muscles or those that are spastic, those that are having rigidity, those that cause rigidity to loosen up a little bit. Fortunately, this is a very crucial step in the process of rehabilitation of these patients. Unfortunately, it turns out that this is only temporary. After some time, the rigidity will return, as in this lasts for some weeks, few weeks. After some time, then they will have to take one more dose of Botox, right. The details of how this is used is beyond the scope of this class. Those who are interested can check the clinical and cosmetic use of Botox or botulinum toxin. So, Botox is the short form for this toxin. It is possible to synthesize and isolate this toxin in the lab, which is then used for rehabilitation and cosmetic purposes. Those who are interested can check what are the cosmetic and clinical applications of Botox, can check. Now, so far what we have seen hemicoline, we have seen Vesamicol, we have seen botulinum toxin, and there are a few more. The other one is tetanus toxin. Tetanus toxin is tetanospasmin that is produced by the bacteria C. tetani or Clostridium tetani. This is one of the most potent toxins known. Actually, it is the third most potent after Botox. Basically, there is Botox, there is diphtheria toxin, then there is tetanus toxin. These are the order of this. You need a little bit more than 2 nanograms per kilogram for this to be very potent. Right. So, what is its action? What this does is it blocks the release of inhibitory neurotransmitters. So, what happens in the neuromuscular junction, right? Once acetylcholine is released, some acetylcholine may remain in the system. For it to stop acting anymore, acetylcholine esterase breaks acetylcholine into choline plus, we saw this, acetylcholine esterase breaks acetylcholine into choline plus acetate, but its action is also stopped by other receptors that take GABA. Whenever GABA enters into the picture, it hyperpolarizes the postsynaptic membrane, right. This way the effect of acetylcholine is neutralized. This is needed in real life everyday movement, this happens in the healthy case. So, this inhibitory neurotransmitter, if it is blocked, then what happens is acetylcholine is present for a relatively long period, causing rigidity, spasms and unwanted movements. Okay. So, tetanospasmin essentially blocks the release of inhibitory neurotransmitters such as GABA and prevents them from neutralizing the effect of acetylcholine when it is not wanted. The action of acetylcholine is balanced by the inhibitory neurotransmitters such as GABA and the action of esterases such as acetylcholine esterase and other such things. So, when this fine balance is tipped or disturbed in favor of acetylcholine, what happens is excess acetylcholine remains at the postsynaptic terminal, causing unwanted movements, causing spasms, causing rigidity, etc. Okay. So, this leads to a situation where there is a spasm or spastic paralysis. Let us remember what happens with the case of Botox, right. It causes flaccid paralysis. This causes flaccid paralysis, whereas tetanus toxin or tetanospasmin actually causes spastic paralysis. There is a group of aeropoisons, there is a group of drugs called curare toxins, right, that are sourced from, this can be sourced from plant and animal sources, from plants such as Strychnos toxifera, curare toxins are produced, but specifically from a plant called Chondrodendron tomentosum, tubocurarine or more specifically D-tubocurarine chloride is produced. What this does is this competes with acetylcholine for space in the nicotinic cholinergic receptor. Basically, it blocks the nicotinic cholinergic receptor from accepting any more acetylcholine, it blocks this, right. So, when acetylcholine molecule reaches this point, it cannot attach to that point because already tubocurarine has attached. Because of this reason, acetylcholine will remain in the synaptic cleft. Until and unless acetylcholine attaches to the nicotinic cholinergic receptor, the channel will not open and sodium cannot enter inside, leading to a situation where there is flaccidity, weakness, paralysis. Actually, when it reaches the lung muscles, breathing muscles, it could stop breathing and essentially cause death. This is how aeropoisons are used. So, basically tubocurarine chloride is coated on the arrow, and it is, when it is shot on the animal or the game of interest or the particular target of interest, this animal is paralyzed by the action of tubocurarine. essentially what happens is that this blocking of the nicotinic cholinergic receptor by tubercularine causes paralysis of the animal, but it is a relatively slow process. It takes about 5 minutes. Its effect lasts for about 120 minutes. For the effect to start, for the effect to take role, it takes about 5 minutes. In comparison with botulinum toxin, in comparison with the other toxins, this is relatively slow poison, right, and its effect lasts for only about 120 minutes approximately. So, these are all approximate values. This is used in South American rainforest by tribes who hunt for animals using these arrow poisons. This is still used in northeastern India, states such as Assam, Meghalaya, and in Myanmar for hunting. It is still used, and of course, it is used in Brazil and Amazonian rainforests, where the tribes of the Amazonian rainforest still use these arrow poisons to hunt animals, right. So, this is the action of that. So, in summary, what are the various things that we have seen? We have seen myasthenia gravis, which is an autoimmune disorder. What this does, it blocks the nicotinic cholinergic receptors in the postsynaptic terminal. So, essentially causing drooping of eyelids, difficulty in swallowing, weakness of the muscle, right. Then there is a Lambert-Eaton myasthenic syndrome. This is again an autoimmune disorder. This blocks calcium channels in the presynaptic terminals, right, causing difficulty in climbing stairs, raising from the sitting position, and because muscles are weakened, right, because of muscle weakness. Then there is botulinum toxin. Botulinum toxin essentially prevents the release of acetylcholine from the presynaptic terminal. How? By cleaving the snare proteins, essentially prevents the action of the docking protein synaptobrevin and syntaxin from fusing to the membrane and releasing acetylcholine into the cleft, right. It leads to total blockade, paralysis, as in flaccid paralysis, and death, right. Hemicholine basically prevents the reuptake of choline into the presynaptic terminal, essentially depleting acetylcholine stores from the presynaptic terminal, and at some point acetylcholine will become empty, or basically at some point presynaptic terminal will not have any more acetylcholine to release and causing weakness. Vesamical, what it does is it inhibits acetylcholine uptake into synaptic vesicles. Basically it blocks packaging of acetylcholine into the vesicles, thus reducing the possibility of its release, right. Essentially this leads to a failure of neurotransmission, paralysis, and when it reaches the respiratory system, respiratory blockade, right. Tetanus toxin, what it does is it blocks the release of inhibitory neurotransmitters, not the excitatory neurotransmitters. It blocks the release of inhibitory neurotransmitters, thus reducing the possibility of neutralizing the effect of acetylcholine. The effect of acetylcholine is excitatory on the postsynaptic terminal, on the muscle, until and unless an hyperpolarization arrives or an inhibitory neurotransmitter arrives, basically GABA or other inhibitory neurotransmitters arrive, there will be some activity in the muscle, right. Tetanospasmin basically blocks the release of these inhibitory neurotransmitters, leading to spasm, unwanted movements, rigidity, and difficulty in stopping the movements. Then you have curare drugs or arrow poisons such as tubercularine chloride, which competes with acetylcholine for basically occupying the space allotted for acetylcholine, which is basically the nicotinic cholinergic receptor is blocked by D-tubercularine chloride, which is an arrow poison, thus preventing acetylcholine from binding. Note, only when acetylcholine is bound to the receptor, the channel will open and let in sodium, not when anything is binding. So, because of this reason, sodium will not enter inside when tuberculin is attached to the postsynaptic receptor, basically to the nicotinicholinergic receptor. This leads to failure of sodium influx and action potential transmission to the muscle, basically leads to weakness, paralysis, total blockade. Depending on the amount, it could also lead to total blockade or death due to failure of respiratory muscles, right. So, with this we come to the end of this lecture. Thank you very much for your attention.